Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PICC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Okay, let's get started because we want to be on time, and I ask the co-speakers to be mindful. Please mute yourself if you haven't done so. If you're not sure where you are, this is a decision summary, lessons from the first FDA approval of an AI tool in diagnostic pathology, jointly organized by the groups that are portrayed here on this slide. And I wanted to take 1 minute to briefly introduce one of the co-sponsors / organizers, and that is the Pathology Innovation Collaborative Community. We are a regulatory science initiative with the following aims. If you haven't been at our website, pathologyinnovationcc.org, you can find all of the information, including this session later on, as sort of an outline. Our main aim is, of course, to do regulatory science, and you can see we do some interesting things. For today, there are 324 people registered, so thank you for that. You see the breakdown on this little pie chart, heavy industry focus, academia, but also patient advocacy, regulators, professional organization, and some other groups. We have to state one important component, which is that this fully aligns with antitrust monitoring. So we can't discuss those 6 points that are listed here, pricing, sales territory, customers, etc. But it is our distinct pleasure to have some of the people in the industry present here. As a starting point, what we provide with Pathology Innovation CC is an infrastructure to connect all stakeholders. And this is one of the sessions. Again, if you want to find out more, I defer to the website. Esther, do you want to briefly introduce DPA?
Yep, sure. Thank you, Joe. So welcome everybody, and I think this is a great opportunity to be here. And also what the DPA is doing, our mission is really about focus on facilitating awareness, similar as to the PICC, of course, education and adoption of digital pathology. And as you can see on the right side of the slide, that's mostly what we're focusing on. We have different working groups, working, for example, on regulatory standards, but also we have the DPA community collaboration group where you can also share experiences on digital pathology. So having said that, can you move to the next slide, please? So What I would like to point out is that when I joined the DPA and also when I started to work in digital pathology, what I noticed is that we have to keep it simple. And that's also what you see in the PIcc, and everywhere that we're working with the DPA, we take it step by step, we break it down in whatever we can to facilitate this and to help you moving forward. So I think what I do want to mention here, that digital pathology is a key component of digital health. With the advancement in digitology, computational pathology, and artificial intelligence, where we are right now, it's more important that we have all these communities collaborating with each other. So we have many hurdles to overcome, many aspects, but the goal remains unchanged, and that's patient diagnosis. All the stakeholders, well, that's my belief, all the stakeholders do want this, and therefore I'm very grateful that we have this session today about the decision summaries, which goes from like what entails like tissue preparation all the way to, for example, automated or an aid in diagnosis to pathology. With that, I'll give it back to you, Joe.
Perfect. Thank you. Thank you so much. So to set the stage for today, I think you've seen probably some of the publications, and we recently tabulated the list of FDA approvals with an AI-based algorithm. You see we're by now probably over 80. So this is a field that's of course of great interest. And when it comes to FDA approvals, you know, there are many different pathways. This is one of the many diagrams that you are probably familiar with or not. But one layer when it comes to the final decision, so in this plane, is that there are certain authorization documents, and one that we want to highlight today in relation to AI, probably, you know, the first AI tool for digital pathology, is one specific document, and that's called the decision summary. And it's my great pleasure to hand it over to Peter Yang. He's the de Novo program lead and works specifically around decision summaries. Peter, pleasure having you.
Hi, good morning. My name is Peter Yang. Yeah, I'm the program lead for the de Novo program. I provide programmatic support and technical oversight and regulatory oversight for all de Novo requests reviewed in the Center for Devices and Radiological Health at the FDA. Next slide. So let me just start with just briefly talking, what are we talking about here when we talk about decision summaries? And just, I understand that not everyone is necessarily familiar with FDA as an organization. So I just wanted to briefly cover this to make sure that we have background. And by the way, I'm packed to the gills with information here, so I'm going to try to speed through these slides pretty quickly. So of course, FDA, sorry, you go back actually, thanks. FDA assures the safety and effectiveness of medical devices, including IVDs, including devices for use in pathology. Of course, we base our decision-making in predictable and least burdensome requirements. So we adopt a risk-based classification process that kind of governs the requirements needed for how devices can actually get into market. But at the same time, we want to be transparent about how we make our decisions, provide context for future device manufacturers and submitters, so that they understand what we're doing and why we're doing it. With respect to PIcc Prostate specifically, which is, we have experts, we have folks from that company here. PIcc Prostate was granted through a de Novo request that was mentioned on one of the previous slides. And as with other submissions, de Novo requests have decision summaries about how FDA came to its decision. And so what I do is I help manage that program. Next slide. So if you don't know what we're talking about, you can actually just, it's probably just easier if you just do a web search for it. So if you search for FDA medical device databases, if you get to our de Novo database and you search there for PIcc Prostate and you click on the decision summary link, and if you just click on that link there, well, the slides will be available later. But if you search for that kind of information, you can pull up a PDF document of the decision summary for this document. You can follow along as we're discussing this, this submission. Next slide. Yeah, thanks. So the decision summaries will vary depending on what kind of submission you're sending forward. So, the big three categories of submissions, I would say, are 510K pre-market notifications, de Novo requests, and pre-market approvals. And the names are going to be slightly different there, but the meaning is going to be effectively be the same. The purpose of these decision summaries is to not only provide transparency into our decision making, as I said, but then to serve as a comparison and as a reference for future submissions so that you know what's been done and what could be done next. So PIcc Prostate was granted as a de novo request, and what we did was we actually created a new regulation under this regulation here. So this is actually enshrined in law. These devices are all categorized under what's called 21 CFR 864 3750, software algorithm device to assist users in digital pathology. So as you can see there, the regulation is fairly broad. It's not specific to PIcc Prostate. And basically, this is FDA's signal that we're willing to entertain other kinds of software algorithm devices, devices with AI, to provide assistance to users in determining pathology diagnoses, to evaluate whole slide images, provide information about relevance of interesting features or locations, and to and all of those devices are welcome under this reg. Next slide. As part of creating the regulation, we also create what's called special controls. Special controls are legal requirements for all devices in the regulation. They are written into the new classification regulation itself. So what I didn't show you on the previous slide was all the special controls that are listed there. And if you look at the decision summary, if you look at the granting order, you can see the special controls that are listed for this document, for this device and for devices within the regulation. Special controls can include things like non-clinical validation requirements, analytical validation, clinical validation requirements, labeling requirements, and the de novo device has to meet its own special controls. So we're reviewing this, we were reviewing PIcc Prostate, reviewing the data for it, and at the same time constructing a paradigm for how future devices would be reviewed. And that paradigm includes legal requirements for the kinds of information that need to be provided, need to be collected for the device, and ultimately the FDA needs to review in order to permit marketing of future devices with software AI and so on. The decision summary, what it does is when you combine it with the granting order, and by the way, if you go back to the database there, you can find the granting letter link there as well. Those two things together tell FDA's risk-based classification story. So from the new regulation to the risk mitigation table, to the special controls, to a device description of what the device is, to the non-clinical and clinical data summaries that were provided for the device to support its granting, as well as our benefit risk thinking and some insight into how we came to our decision overall. All of that information is available in the decision summary. And so as you read through and understand how this regulation was constructed, it does a couple of things. One is it demonstrates how our special controls were met. So you might read the special controls, which again are legal requirements, and you might say, well, I don't understand how I'm supposed to provide data to meet these. What exactly should I be doing? One way to get context for that would be to see the decision summary, look at the data summaries, look at the risks and how we tailored our requests and how those requests for data were met with information that PIcc provided to us. The other thing that this decision summary does is it serves as a reference to support future 510K submissions. So one of the things about a de novo request is after the de novo request has been granted, future devices are actually submitted through the 510K process. And unfortunately, I only have 10 minutes. I don't really want to get into all the sort of nuances of 510K submissions and so on. But basically, if you have an AI system for, maybe not prostate cancer, but some kind of other cancer, and you say, okay, well, I want to compare my device. I want to get my device onto the market. One way that you could do that is you could potentially demonstrate that your device is as safe and effective as the PIcc Prostate device, and that's the sort of 510K review standard. And so in doing that, you can review the decision summary for PIcc Prostate, understand what data was collected there to support the granting decision, and then say, okay, here's what I need to do for my own future 510K as well. And I can do a similar kind of test. I can do similar kinds of comparisons or analyses, or include similar kinds of labeling requirements. And that's what the decision summary is good for there. Next slide. So when it comes to decision summaries for AI, and I'm going to stick to a very, very high level description here, generally we would recommend that a decision summary discuss how the AI was developed, or how the model was developed, the data set that was used to train the model, the validation process and the data set that was used to validate the model for real world use, and then things like warnings or precautions for how to use the AI software, things that the AI software is not designed to do, for example, and then any information needed for ensuring correct use. What kinds of information does this AI software expect? How should information be processed? And what kinds of results should I be getting? And again, limitations with those kinds of information. Next slide. And so this summary slide sort of sums it up in terms of what I've been talking about already, right? So with the decision summary, you can understand what the sponsor needed to do to get their device granted or cleared or approved. You can understand how the sponsor met the special control and why we requested certain information, what kinds of risks we were trying to address. If you have your own testing strategy, you're developing your own AI software, then what you can do is you can align that strategy to meet FDA's requirements, understand what FDA is asking for, and ultimately, you know, and I would strongly recommend this, assemble a testing strategy document, right? That talks about the kinds of testing you're going to do, how you're going to develop your AI system, the model, and so forth. And then, you know, start conversations early with FDA. So in the context of a pre-submission, which is basically, you know, before you submit your actual marketing application, submit a pre-submission and sort of get some feedback from the FDA as to where your software is going to go and what kinds of concerns the FDA might have, and then tailor your stuff to match that when you're in the context of the, you know, an actual medical device submission, you sort of got all of your stuff together. And that's it. My email is there. If anyone's interested in asking questions directly, I'd be more than happy to take your questions by email as well. Thanks.
Thank you so much, Peter. I hand it over straight to Emre Gültürk, the Vice President of Regulatory Affairs and QA at Page. Emre?
Thanks, Joe, and thanks, Peter, for setting the stage. So after we heard hearing the agency's perspective on decision summaries, I think it will be beneficial to provide the industry's perspective, right? Especially being the first-hand experience recently interacting with the agency, I'd like to go over and just strictly focus on the industry's perspective here. Yeah, thank you. So, not to state the obvious, but the decision summaries are obviously a very high-level output from a much larger submission effort here. So, just to provide a rough number, we roughly had 2,000 pages of documentation exchange with the agency as part of the review process. And as you see, decision summaries at 20 to 30 pages, high-level summary, just highlighting the key aspects, which we're going to go over in a second. And from our perspective, really its main goal is to providing the obvious and key details to the predicate device manufacturers. Again, in this case, this could be ourselves, right? As if we did our second product, we would also look at the similar public information here, even though we have more internal details. But for another company, obviously, they would access these details, which are publicly available, to understand the key elements where they need to assume and start developing their materials to start engaging with the FDA. Again, so based on our experience with the agency, what the level of detail that they're striving for is enough that they're avoiding unnecessary pre-submission rounds to potentially lower the burden on the agency's end as well. Again, just to give an example here. So if an industry manufacturer approaches the FDA and just to ask a focused question around, should we assess the standalone performance of our model, FDA does think that that's potentially an unnecessary level of question, which they aim to resolve that with providing details in a decision summary. So hopefully when a manufacturer approaches the FDA, they would know that and hopefully come up with a specific testing plan to discuss appropriate to their device outputs. So that then there could be a potential fruitful discussion with the agency so that they can provide a specific feedbacks, understanding the differences, what that device provides in terms of the output, and kind of tailor, work together and tailor that testing plan to help the manufacturer to move forward. So that's kind of simply what's been our experience with the agency, just striking that right level of detail, but at the same time keeping, again, not providing an exact prescription where every company would follow, is keeping those, holding those details so that it would encourage the manufacturers to approach the FDA to discuss, confirm, or iterate and reconfirm, so that, again, that's a fruitful and a useful discussion. So that's been our understanding from the industry's perspective. Next slide. So I'd like to break down the decision summaries into a few key components, again, from our understanding. One of the most important aspects in a decision summary is the intended use / indications for use. And the reason for this is that the regulations are pretty clear. When you do certain modifications to an intended use, you do need to go through a pre-market review to confirm that the modifications, via under a special 510K or traditional, again, depending on the scope here. But ultimately, our interaction with FDA and our confirmation here is that FDA is interested in reviewing these modifications. Hence, they were very specific in including certain elements into our intended use and indications for use, which I highlighted here, such as the tissue type, such as the specific output that the device generates. In this case, it's an XY coordinate on a location. And the compatibility aspect of the device. Again, so the PIcc Prostate in this instance is compatible with the Philips ultrafast scanner and the Page's full focus image viewer. So they'd like to lock these configurations down. When you, for example, have an additional FDA cleared scanner that you'd like to add in your compatibility, it seems to be that they'd like to review that data before you can just automatically go and update your intended use. So that's the intention here for them to, at least our interpretation in terms of the intention here, to include these statements in the intended use so that these modifications would trigger a review on the agency's end. And obviously the use setting, as I mentioned, such as being a diagnostic aid, adjunctive use setting here, they also would like that. So ultimately, it's making it abundantly clear that it's not an autonomous AI or anything like that. So that, again, those limitations are clearly outlined in the intended use statement. And another key component that I like to highlight in decision summaries are the development data set, its distribution and its diversity. Historically in radiology, there's been some backlash and some conflicts in terms of the diversity of data in the training data set. So FDA was intentionally more careful in our instance, at least our understanding is. So they specifically vetted the development data set and understanding the diversity of the data set in terms of the race, ethnicity, age distribution, could be challenging scenarios, mimickers, and a variety of different aspects to make sure that, again, from the beginning, system is designed and developed that can inherently generalize and ultimately not specifically in a given validation effort. Next slide, please. Another important aspect in the decision summary is the mechanism of actions that are outlined and the principles of operation. So again, the decision summary sets the setting by starting with the intended use and ultimately switching to this by explaining that how the device should be used. It's very clearly outlined in the decision summary that, again, making it clear that it's not an autonomous AI. It should first, the pathologist should conduct their current standard of care review. They should then look at the PIcc Prostate in this given instance, and then they should make their decision, right? This helps the public and the following device manufacturer in terms of when a study is designed, it is designed appropriately to this mechanism of action and principles of operation. So that when they have a use case that might be different than this, again, so they should consider that potentially those aspects are impacted so that they need to interact with the agency to confirm what those inherent differences can impact on their testing plans. So this is simply what it provides to the industry, to the manufacturers, as well as to the public in terms of the devices used. Next slide, please. And another important aspect is obviously the performance characteristics. Again, so also outlining the study data characteristics and its diversity is an important factor, just like we just talked about the development data sets, characteristics and diversity. Obviously, it's public information to provide this to make sure that the data that the performance characteristics are reported on are representative, challenging enough, again, diverse enough, again, age, race, ethnicity, challenging scenarios, mimickers, variety, whatever it may be. That ultimately it holds a strong representation to be able to challenge the device, but also it is adequate in terms of the sample size, and all of those stratifications can happen to show that system generalizes in challenging scenarios as well. So again, so we have results in our decision summary from the slide level accuracy all the way into localization, segmentation, precision of the device between multiple operators or multiple scanners, how the system generalized. So a given slide can be scanned multiple times by multiple operators. Again, they're trying to replicate an actual clinical use and prove that the system can still generalize so that it doesn't fail from one scan to another for the same slide image, which could be concerning if this was commercially available. So those are the aspects that they're striving for, and those are the aspects they include in the decision summaries to make sure that they're considered for the predicate devices as part of their testing plan. And obviously for the clinical performance studies, a reader diagnostic accuracy study has been summarized in our decision summary. And the details around that, again, I just put an example in there, just a table and an explanation what that table means from the decision summary. But ultimately, a variety of different tables are introduced, and those are the key outputs from these efforts per the agency and per page, per industry, with an alignment that those outputs are valuable to the industry and the public to review and understand the strengths, and again, realistic limitations of the device as well. Because I just want to make it very abundantly clear. Device do work when it's strictly followed to its intended use, but industry needs to understand that the use setting, the compatibility, and anything beyond that can introduce certain limitations so that everyone is very clear understanding that when FDA does approve something, it is strictly within the boundaries of its output, the compatibility aspects, and whatnot. So those are kind of complemented with these performance characteristics so that the public can interpret what the device can and can't do. Next slide, please. And obviously, another important aspect for the decision summaries are the benefit risk determination. Again, this is a joint effort by the submitter and the agency to come to an agreement, making the case that the benefits are favorable compared to the risks that the device presents. And this is obviously publicly posted so that the individuals can understand what the device brings to the table with the outputs that it presents, and what potential risks that it brings in the AI world is very obvious, such as false negatives and false positives being the most obvious ones. But kind of how the risks are potentially mitigated. And again, communicating that back to the predicate manufacturers as well as the public to consider and understand the strengths and potential limitations of the device. Next slide, please. So Peter did go over this overview, so I'm not going to go into the detail, but one thing that is not necessarily directly in the decision summaries, but is the next link as part of the de novo approval is the reclassification orders. They do include the special controls until the regulation is publicly posted in the Code of Federal Regulations database. What that means is that when FDA does establish a new product classification, that product classification comes with very unique special controls that each predicate manufacturer needs to review and adhere to. So it provides very, very important guidance. Again, it's not part of the decision summaries, but should definitely be looked at for the intended use, what the specific intended use statement includes, right? The specimen type, the compatibility, the use setting, all of those things are specifically called out in the special controls. The device label, the device limitations, what it can and it cannot do, how it should or should not be used. Details about its performance testing, again, leaving the door open, saying that the analytical and clinical testing at FDA's discretion should be discussed and confirmed. Like all those things are kind of laid out in the special controls. And ultimately, the specific testing plans, right? So certain inherent details are also included in the special controls where a predicate manufacturer needs to review and adhere to, so that when they approach the FDA, those are considered and potentially incorporated into their materials for the agency's review. So with that being said, I think that's all on my end.
Perfect. Thank you so much, Emre. This is a great overview. I hand it over to Dr. Jansen Seeholt. He's a member of the Machine Learning Working Group for CAP. Jansen, please take it away.
Can you hear me?
Yes.
Okay. We can skip to the next slide. So I have to give a very brief overview of how the laboratory can actually use the information in the decision summary to both evaluate device or software, as stated, and also compare the claims that the manufacturer or vendor provides to how the device or software performs in a local setting. I'm going to be sticking with CLIA, CAP, and ISO terminology, which is verifying the manufacturer's claims for deployment in your clinical laboratory. Next slide. So there's a lot of useful information in the decision summary. Much of this has been summarized already, such as the intended use, the indications for use. I'm going to stick to the performance claims that the vendor or manufacturer makes, and also something else that we can glean from the decision summary is how we can actually design a verification study similar to the vendor's design. Next slide. So we saw the indications for use. I'm going to be using the PIcc Prostate needle core biopsy whole site image analysis software as the case study in this short presentation. It's an adjunctive computer-assisted methodology that we covered before. Next slide. Some things that you might want to understand from the decision summary is the volume and complexity of the training, tuning, and test data sets, and Emery covered some of this already. Interestingly, in the test data set, they had more than 11,000 slides prepared at more than 200 external sites, but diagnosed at a single internal site. And then you might also want to understand the complexity of the patient population and how that mirrors or mimics the patient population that you see in a routine clinical care. So for PIcc Prostate, about 80% of the slides were collected from Caucasian patients, about 8 to 9% from Black or African American patients, and about 3% from Asians. Next slide. The key information that I would glean from the decision summary for laboratory verification purposes would be from the accuracy study and the clinical study when I'm trying to verify the accuracy characteristic claim put forward by the vendor or the manufacturer. So in the accuracy study, they had 311 cancer slides, 417 benign slides, and the claim is a sensitivity of 94.5% and a specificity of 94% for predicting whether it's cancerous or benign. This, of course, does not take into account localization accuracy, and you might have to evaluate that separately. Then they also had a clinical study where they used 171 cancer slides, 356 benign slides read by 16 pathologists, and they came up with these assisted macro average values for sensitivity and specificity of 96.8% and 89.5%. And then you can look at the case breakdown. They had about a 50-50 breakdown for tumor size less than or equal to 0.5 millimeters versus greater than 0.5 millimeters. And then you can also look at the case breakdown for benign tissue specimens and whether that mimics your local laboratory environment. So I'm going to focus here on the accuracy metric, which is 94.2%, and how we might verify that metric when we're trying to deploy this algorithm in the local laboratory environment. Next slide. The other set of useful claims that you can obtain from the decision summary are the precision characteristics and also how the vendor actually did the precision study. Because this might be very helpful when you try to come up with your own verification plan for a precision study. So for repeatability or within scanner precision, they took the same slide and scanned it 3 times using the same scan and operator. And then for the reproducibility study, they took the slide, scanned once with 3 different scanners at different locations by 3 different operators, 1 operator per scanner. And you can look at these metrics at average across all slides or per slide. So for example, for the repeatability study for benign slides, about 94.4% of all scans produced the right result, whereas 88.9% of all slides produced the correct result. Next slide. I'm not going to go into the details of this, but how you might come up with the sample size for your local laboratory verification can use a very straightforward power-based sample size calculation. And I'm going to focus on the 3rd bullet point. For example, if the lab wishes to compare the locally determined accuracy of a software to the manufacturer's claim of 94.2%, the sample size required to have 95% confidence and 80% power to detect a difference of 5% lower than a claimed accuracy of 94.2% would be a sample size of 166. Excuse me. Next slide. You need to think about things like the balance between cancer versus benign slides. These may not affect the sample size for an accuracy metric, but they do for sensitivity and specificity. And of course, if your data set is not similar to the manufacturer's data set, that might actually explain why you failed to verify the manufacturer's stated claims. Next slide. So this is just a simulated accuracy study using 166 samples of 50-50 split of cancer and benign. In this simulated study, we compared our race or ethnicity breakdown and also the other characteristics compared to the manufacturer's data set. And we did not see any statistically significant differences. We have an observed accuracy of 91.6%. And even though it's lower than the manufacturer's stated claim of 94.2%, simple one-sided test of one sample test of proportion will show that this is not significantly lower than the vendor's claim. Next slide. You could design a similar precision study, a simple precision study where you take 10 slides, scan 3 times using one scanner operator, or a reproducibility study using 10 slides scanned once with different scanners at different locations by different operators, and compare your performance to the vendor's claim to see whether it matches. Next slide. So those are the claims that we would extract from the decision summary and how we might formulate a verification plan. Of course, this is not the full extent of your responsibilities as a laboratory director. You might also have to look at things like reportable range and reference interval, depending on the method or the software that you're verifying. And you should also always try to break the model to try to understand its limitations when it fails, such as like a failure in effect modes analysis. That's it, I think.
Thank you very much. This was a fantastic summary. As the last planned speaker, it's my pleasure to introduce Dr. David Klimstra. David? I think you're muted.
Sorry. Thanks very much, Joe. So I am also at PAGE. I'm the Chief Medical Officer at PAGE. And I'd like to broaden the discussion a little bit to talk about what this decision summary and approval means for the future of AI in pathology. Next slide. If you look at this product classification statement, and this has already been referenced earlier, we see that as Peter said in his presentation, this is a very broad device classification. It applies essentially to any software algorithm to be applied to assist in the diagnosis of digital whole slide images. And so this enables the same classification to be applied across a range of other AI tools that may be developed in digital pathology. And so this approval basically establishes the predicate for other approvals, which can now be submitted as a regular 510K rather than going through the de novo process as Paige did. Next slide. So what does this mean for our industry? This establishes in our view that you can confidently use AI-based devices to help pathologists arrive at the correct diagnosis, what we call decision support, to help in the analysis of digital images and achieve an accurate diagnosis. Also, very importantly, it ensures that the AI, in this case, generalizes across practice settings and laboratory variables based on training, as highlighted previously on slides from many different technical laboratories that were included. And so that one can confidently use this product, knowing that it will be generally applicable. Very importantly, this creates a pathway for clearances for future products in the category, and sets an important quality bar for future products to ensure that they meet a certain level of regulatory rigor. It's important to know that there is interoperability here, but it is limited to an FDA cleared devices. And so if you look at the full process that Emery briefly showed of how this is used, it's used in concert with FDA cleared scanners, viewers, monitors, etc. And even within FDA cleared devices, expansion to other FDA cleared devices would still require additional 510K pre-market review. So it is important to understand that each expansion will require additional data to be assembled and reviewed by the agency. And it was clear to us that the FDA intends to enforce these regulations rigorously. Next slide. What does this mean for pathologists? I think this is basically signing a definition for clinical grade AI. AI that has been rigorously reviewed and approved for use as an aid to pathologists. And this enables additional scrutiny for slides, giving pathologists more confidence in their diagnosis, and allows them, importantly, to focus their attention on the most critical aspects of establishing the diagnosis, knowing that they have this sort of decision support for the screening component. The compatibility is the responsibility of the manufacturer, as is the quality control. And that's obviously very important if you compare something, for instance, to a laboratory developed test where all of that rests with the individual laboratory. And this also allows us to, or the laboratory to scale use. If you have a multi-site laboratory, you can undertake a single verification and then use the product in all your sites, as opposed to laboratory developed tests, which require independent CLIA validation for each physical location. And the verification that Jansen described is actually quite a lot simpler than preparing your own laboratory developed test. As I mentioned previously, these cleared devices are locked down. I think there is perhaps some misperception in the field that when you use AI, it literally learns as you use it and evolves. But that's not the purpose of an approved device. An approved device is locked down, and any modifications will require FDA re-review in order for them to be incorporated. So the device is what it is, as established by this clearance. And then furthermore, modifications of the end-to-end test will require an LDT for that specific modification. So if you decided to use this, for instance, with a different scanner that had not been FDA cleared, that would require an LDT. Next slide. And then finally, just very broadly, where are we headed now? And I think, this sets the stage for the development of a lot of other pathology decision support AI for use cases where it may be helpful in clinical practice. We now have established processes to evaluate the performance of these algorithms, and as I said, to ensure that they generalize on different laboratory settings, on different technical preparations. The FDA has essentially established clinical grade quality, and using FDA cleared devices obviously eases implementation for the users. And then there's this nice feedback loop that as you increase the availability of decision support AI, it will prompt more people to move into the digital environment, which is something that we have to acknowledge has been rather slow in pathology. But having access to tools that enhance the performance of the digital workflow should push more people to move into the digital environment. The feedback loop is that the increasing use of digital pathology then opens the door for the development of more advanced computational tools to further enhance our ability to function in this environment. Things such as digital biomarkers or multimodal data analytics using data from other high-powered assays. So we think that this is a major step forward in facilitating the transformation of pathology to the digital environment. And that is my last slide, so I'll turn it back to Joe to moderate for the discussion.
Wonderful. Thank you so much, David, and thanks to all the speakers. I think as a next part, I want to say thank you to Ola Green for organizing all of this, and also introduce Brandon Gallis. He serves as our liaison with the Pathology Innovation Collaborative community. Brandon, are you on?
Hi, Joe. Yeah, sure, I'm here.
Perfect, because I think it may benefit, being in the Office of Science and Engineering laboratories. Some of the questions I see are very detailed, so I just wanted to call you out directly to have you as another expert from within the agency. So I think probably many of you have followed some of the questions. We want to go through those because these are really, really interesting. So again, we get a lot of questions whether we share the slides. Yes, everything, including the recording, will be on the website. The first question is from Alice Guinea. Will the FDA be defining consensus standards for PIcc prostate device product code?
So this is Peter. I can try to take some of that question, and maybe Brandon, if he has anything to add, can add on. So I don't know what the agency's plans are for this area. I would say that I think what you're referring to would be something like a guidance document that FDA would put out that would help provide some explanation about, maybe how AI systems should be developed and validated. I think there's other questions about what would FDA's expectations be for certain kinds of testing. And that's where, generally speaking, a guidance document would be the best way to do that. It would be something that we could update with best practices. Again, that's just me as an individual sort of thinking what would be the best approach to doing that, but I don't know the agency's plans in this area. And as you can imagine, for different kinds of applications, your AI system might have to be more robust or less robust. And so we might have to, getting into the nitty gritty of those kinds of recommendations could be a little bit complicated. And I think, this area is definitely an emerging field. And so I think we're trying to step thoughtfully and carefully through all of that. And so right now, I think the way that we're, working is not necessarily putting out guidance, but certainly considering each device in its own individual context and its intended use, and kind of, titrating the calibration requirements there, so that we understand, we make sure that we have a sense of the device being, analytically and clinically validated for its purpose.
Maybe as a follow-up to this question from Joy Kavanaugh, Peter, you very rightly recommended early engagement with the FDA via a pre-submission process. Can you comment on current wait times for pre-subs, and specifically, has this returned to normal availability similar to pre-COVID or still limited?
Yeah, so this is changing. It actually has already changed. So there are some limitations. Previously, the in vitro diagnostics group did not actually accept pre-submissions for anything not related specifically to the COVID-19 pandemic. So as of June 1st, the IVD group is now accepting pre-submissions for all IVDs. However, those may still be reviewed on an extended timeline. I can tell you just from within the agency, the IVD group has been working day and night for 2 years. And they've been working really hard on all the submissions coming in. And so I think they're trying to balance all of their work and sort of make sure that they can try to meet expectations, but that's our current expectation right now. So it would be an extended timeline, but we will accept your pre-submission as opposed to saying we just have to deprioritize this for now. And I'm hoping that that will, continue to facilitate those kinds of discussions within the agency. But again, maybe on a bit of a delay.
Perfect. Next question, Alice Guinea also asked, thank you. Could I also, if Peter or the FDA, talk to the purpose of defining the image viewer in the intended purpose?
I can't speak to that. In particular, I think it's probably the pathology group wanting to, have some level of control or some level of assurance over sort of the entire pipeline to make sure that everything's being displayed in a rigorous way. But I don't have further information on that. You would have to talk to the pathology group to find out more about their specific, requirements in that arena.
So I would say that the viewer, it opens the image and provides the pixels to the algorithm. So to make sure that that process is expected by the algorithm, there's one method, at least for now, is tying it to the viewer. I think that the relationship between the viewer and the image is so important that that needs to be ensured for now. These are my opinions. And then also those images all have different formats. I guess that's the same point, is making sure, and plus the viewer also is part of the regulatory device. The scan system has the scanner, the image management system, and the display right now all as regulated devices.
Thank you, Brandon. Steph Hess asks, why was the approach as an aid afterwards, after initial diagnosis, and not a guide ahead of final diagnosis taken, I presume? Don't know if that's a question for Paige.
Go ahead, Emery. I see you unmuted.
Absolutely. So I can definitely provide what our interpretation here is, why we, agreeing with FDA, chose to go this way. And again, so it's a brand new industry, and my past experience from digital health, digital therapeutics, AI and radiology, the first products tend to be a little bit geared towards a bit more conservative setting until we can produce enough post-market data to prove that the device is safe to use. So I believe that's been the approach here, again, with FDA, that they simply felt more comfortable in a adjunct setting to collect some post-market data before they can start becoming more comfortable on concurrent use type of settings. So that's why, again, this being the first product, just being on the bit more conservative side.
Can I jump in on that? The indications for use are the vendor's responsibility. The manufacturer is responsible for picking that path. Now, of course, the indications for use also drive how it should be validated. So while I heard that this was an FDA decision, I'm sure it was a back and forth about how they're validating the device, and that the kind of conversation resolved on after the fact, because the indications for use are the industry's responsibility. And then it's about having appropriate validation for that indication for use. And some indications are easier to validate with different, the indications drive the studies.
Exactly. I mean, sorry if that came up wrong. So what I mean is that, yes, theoretically, anything is possible. You could go with the most autonomous AI proposal in the world, and that would come its consequences of a validation that could probably take 10 years to do, right? But still theoretically possible. What I'm trying to say is that keeping it as a potential class 2 and the risk level, obviously those conversations with FDA ended on a threshold where it would keep it on a potential class 2. And again, it's safe enough to start collecting some post-market data for us to even understand the real safety profile of this product.
So one question probably related to this and, the, let's say, intended use is coming from Jennifer PIcc. Is there a recommended sample size going forward for other AI submissions, including for non-cancer AI detection? I mean, you kind of alluded to that already, Brandon.
Yeah. Every disease and task is going to have a different baseline performance. What is the performance happening right now without the AI device? And that baseline kind of drives our thinking about what is the consensus performance level it needs to achieve. In fact, that's often the comparator, is the current performance is a comparator for the performance with the AI in a reader study. So we really try and tailor these to the task, and certainly the risk also plays a role there. So there's no blanket statement for that question.
Yeah, totally agree with Brandon in that. I think if we were to try to develop standards for any kind of AI software for any application, that's where we would get more conservative, right? And I think that would be not necessarily least burdensome for every AI application.
I just have a question. Do you actually do a sample size calculation, or is this like a rule of thumb that you use when you come up with these study samples? Like, do you use a precision-based sample size calculation that takes into account both the expected claim plus the precision that you need around that claim?
So in my research, I work on methods to use pilot studies to size a pivotal study. And certainly that is something I always bring to the conversation when I'm involved. And I know a lot of my colleagues do too. Now, there are expectations to have justifications for sizing, and those we drive it to be a statistical justification, though not every manufacturer takes that kind of approach. And in the end, you know, the size of the study will be a roll of the dice by the manufacturer. But good science says collect some data, understanding the pathologist variability alongside of the biological and case variability. And those two elements feed variance, understanding of those variance components and sizing of a pivotal study. And there's some good work out there under the label multi-reader, multi-case analyses, mostly arising from the radiology world, since radiology has been a little bit more active on these pre-market diagnostic type studies.
There's a two-part question statement from James Harrison. James, thanks for joining. First is, if the local verification with 10% positive cases yields different accuracy from the vendor data with about 50% positive cases, what does that mean? And the second part, I also read this, is the vendor can correctly claim that a significant difference in accuracy is expected with changes in class balance, but that doesn't provide local guidance on whether a particular performance level represent verification or clinical acceptability of performance. So I think this goes to very close to what verification should accomplish and how the performance is assessed. I don't know, Jensen, Brandon, who wants to take?
Jensen's answer to that, and really accuracy is not a general metric we use around here because it pools the two different classes, the diseased class and the non-diseased class, and that class imbalance obfuscates the actual performance on those two populations. And so we always ask for a performance on the diseased cases and performance on the non-diseased cases. And when you have a human in the loop, you have to present those without telling them, of course, and the sensitivity and specificity from humans is something that gets a little bit confounded. And at least in the radiology world, ROC studies are really what we look for as the top level endpoint, because it can average over those thresholds that individuals make on diseased and non-diseased. But there's, you know, every division will address this differently, but the main bottom line is you can't look at accuracy by itself because of that confounding issue of class balance. We always ask for sensitivity and specificity.
Thank you, Brandon. Samuel Chan's question, thank you for posting that, has in part been answered, so we skipped that. The next question comes from Arun Nemani. What is the FDA's guidance for models that ideally should be retrained periodically to ensure robustness? Does this require a formal review upon every retrain? Very good question, especially for, you know, continuously learning models.
Yeah, I think, well, even when it comes to continuously learning models, I think, you know, once again, you're sort of fundamentally altering the core of the algorithm that's going to affect the safety or effectiveness, right? And so, you know, experienced regulatory people will know that I'm referring specifically to a guidance that talks about making modifications to your device and when a new 510(k) submission needs to be submitted. You know, I think we're trying to work on how to develop, how to, you know, progress and make changes to your devices, especially when it comes to AI and especially when it comes to these kinds of self-learning models. In general, I would say, you know, we talked a lot about locked algorithms in the study and sort of needing to have a stepwise approach to making changes. And I think generally speaking, that would be the way that we would go. But it really would depend on the individual algorithm, what you're trying to do in the space, what kinds of changes are proposed to be made. And I think that's where a conversation, again, in collaboration with the group that you're interacting with, is the best way to sort of know how to move forward in the area, because it is going to balance depending on the particular intended use and so on, and how your model was developed originally and so on.
Yeah, I just dropped the 2 guidance documents about when to come back to the FDA when you make changes to your device or when you make changes to your software device.
Sorry to interrupt here.
Thank you.
A couple more minutes. I just want to get one last question in for that one. Does the FDA also have requirements for model monitoring in production? So considerations for when a particular model is out of compliance once it's already deployed?
To my knowledge, I'm not aware of a model that we've cleared that is self-learning, that sort of changes on its own. I could be wrong about that. There might be a device that I haven't seen. Certainly, we haven't done one for the De Novo program, which again is for novel devices, of which that might be one of them. But I think that would be our concern, right? Our concern would be that the model changes over time, that its performance would vary, and then you wouldn't be able to ensure safety or effectiveness. You don't have control over the model. So I think some amount of monitoring would be important. As for the right regulatory framework to ensure that that's happening, I don't have expertise on that, or I can't speak to that right now, but that is something that the agency is definitely interested in. And we've been talking for a long time as part of the digital health program.
Yeah, there has been certainly initiatives and frameworks proposed. Right now, I think there's 3 devices that are allowed to continuously make updates. But we've had some change in our thinking from the lawyers in the Center for Devices. And basically right now, devices that are going to have this learning component, this continuous updating, are going to have a special control that identifies and discusses the continuous learning aspect of the device. And right now, like Peter said, there are no special controls like that for any de novos. And so we are currently thinking that the next one that gets cleared will have to be accompanied by a special control for governing that continuous learning.
All right, as posted just in the chat, we will answer all the remaining questions in the chat offline and provide the answers in writing on the website. I want to be mindful of time and thank all the speakers. This was an amazing session. The recording and all the materials will be posted via PIcc, and we share the content also with the co-sponsor DPA and of course all the people on the call. Thank you very much and hope to see you soon back here.
Thank you, Joe.
Thank you. Take care.
Thank you
so much, Joe.
Bye-bye.